on developing climate change indicators for Tampa Bay. And so I'm gonna talk about the context for this project first, and then Ben is gonna give some insight into some of the work that he's been doing developing these indicators. So obviously, you know, we're the TAC, and this is basically where you all sit within our management conference as a program here. So we're in this box here, and PPEP is a facilitator of the TAC, Community Advisory Committee, our management board and policy board. And oftentimes what we do at the TAC is sort of vet some of these ideas, some of these research topics to y'all, kind of get your feedback on where this is headed and if this is a useful avenue to pursue. So that's really kind of the goal for this today. Is this is kind of a prototype that we're showing you all. Are these the correct indicators? Are we tracking the right things? Are there ways to improve this? And hopefully get feedback from y'all. And we actually will have a Google Forms survey for y'all to fill out after this meeting that we'll send out in an email. So as you all know, we at the Estuary program are really keen on various indicators. This is just a quick snapshot of several of the report cards or stoplight graphics that we often call these things that we're currently have methods developed for, and methods being you know, data to synthesis to actual report cards. So we have indicators for water quality, benthic health, fisheries health, seagrass health, tidal creeks, habitat status and trends, sediment health, all of those things. And so a lot of these things are covered in our comprehensive conservation and management plan, our CCMP. And there are certain elements of our CCMP that we don't currently report on, and these are aspects of our program that we care about, but we don't actually have methods to synthesize the data to create a meaningful product that can hopefully produce some actionable outcomes. This is just showing you a lot of the dashboards that we have. So other products that we create, not just report cards or 2-pagers, but interactive components that are really kind of creating a more immersive experience for our management conference. And I bring this up as well because we do have a dashboard for these climate change indicators that Ben is going to show, that is sort of bringing this idea of data to life as a proof of concept. And ultimately what we're going towards, is a comprehensive state of the Bay report card. So there's different elements of our program, of the Bay, of environmental ecological health that we're reporting on, like water quality, fisheries, seagrass, all of these things. But we want to kind of roll this up into an overall Bay report card. We have, of course, our State of the Bay report, which is sort of a narrative description of the Bay. But we want to produce something that is like, oh, well, the Bay gets an A this year or a B this year. You know, and you can debate the pros and cons of that approach. You know, you can say that distilling it into a single number or letter is somewhat problematic because it sort of oversimplifies the ecology of the system. But nonetheless, we still think this is a useful approach to kind of display the overall status of the Bay. And this individual report card is obviously composed of the individual indicators that we have. And this is just an example for the Chesapeake Bay program. Not exactly something we'll copy exactly, but something in concept that we want to kind of emulate. And this is going to be a few years down the line. But of course, before we do this, we have to fill in some of those missing indicators. And one of those that we haven't really done a good job on is climate change. Obviously, it's something that we're all concerned about. We are taking action as a program to think about how we address climate change. One way that we've been meaningfully doing this over the last 10 years or so is actual field-based methods to track primarily the status of sea level rise and how that's affecting tidal wetland habitats. And that's through this critical coastal habitat assessment project right here, where basically at these sites, we've gone out 3 times now and actually done these really comprehensive transect surveys from basically the tidal line upwards into upland habitats to sort of document that habitat and how it's changing. And we have seen some really marked changes in basically mangroves moving upland, which is kind of what we expect, but this is our attempt to really document that quantitatively. But this is like boots on the ground, field work, very intensive, kind of one way to look at how climate change is affecting the Bay. Another way we can do this is sort of report on kind of more routine indicators at sort of a very high level of like, well, broadly speaking, how is water levels changing? How is water temperature changing? How is rainfall changing? How are the number of extreme events changing? Things like that. And this is really just what we see as a gap for just climate change in general. How do we understand these indicators as a broad overview of how it's affecting our region? And that's really kind of the nexus of this project today. We're sort of synthesizing these high level data sets to complement some of these other indicator programs we have that are more detailed at sort of the field-based level. So I'll hand it over to Ben now. That's kind of the context for the project, why we're doing this. Again, we want feedback from y'all. Ben's going to show you the great work that he's been doing so far on this. And we'll hopefully have a discussion about what y'all think about this. So Ben, take it over.
Awesome. Thank you so much, Marcus.
You want
I think
me
I
to
can share. Yeah, actually, if I'll drive it, because I'm going to go through the app. If you don't mind unsharing your screen, then I can share mine.
You got a map?
It looks like I am unable to share my screen until you stop sharing yours.
So I think, yeah, we don't have the settings right, Ben, so I'll just run it from my end if that's okay.
Oh, you can't. It doesn't say that I'm unable to share. It just says that it ha it you have to stop your sharing in order for me to share mine. Perfect. Thank you. Cool. Can you see my screen okay?
We're good, Ben.
Cool. Thank you so much, Marcus, for that great opener, and yeah, just including me in this project. It's pretty exciting to be able to be aligned with the same kind of philosophy of open source project component parts to be able to develop this. So looking forward to getting your feedback. And as far as indicators, real big picture, we're trying to make sure that we're grabbing things that are relevant to both people and the environment or the ecology, and that we're also covering, yeah, not just land and not just sea, but both. The data that we're really interested in, we want it to be publicly available. And we want it to be up to date as well as historical for comparing now versus then. We want it to be updated daily and preferably as close to yesterday as can be. We want it to be authoritative. So who are the entities that are producing the best, highest quality data? And the best possible spatial resolution. And ideally, there's this application programming interface endpoint, meaning that everything can be automated for bringing the best available data into the Tampa Bay region. And we want to build on the TBEP tools and all the great open source products that Marcus has been overseeing, along with many of you others there, and make this open source, and then even other estuaries can reuse these components and apply it to their areas of interest and improve upon any aspect of this. And then with the visualization, we want it to be scientifically credible as well as still accessible to the layperson who doesn't necessarily have a scientific background. And that's where we could really use your help and feedback. Sometimes for us data scientists nerds, we stare at charts too much and assume that they're very intuitive. So it really takes extra sets of eyes to kind of get another fresh take on that. And then we want this to be interactive, both from an engagement perspective with the general audience, as well as to be able to probe the data and look at things that are relevant to anybody within the Tampa Bay area who has a particular interest in a time or place, and then configurable, and we'll look and see how you can change parameters to look at different days or different climatological periods and smooth things out by different numbers of days. So looking at the data sets that we brought into this, Prism seemed to be the best, most widely used standardized air temperature data set that we could get our hands on. It's updated actually 6 different times throughout a 6-month period. And we developed the routines to go and fetch the data for the entire continental United States, and then clip out to the Tampa Bay region, and then organize those files, and then get these zonal statistics. So these are the parameters available, and we're just going to look at the mean temperature, but these are all the parameters available, and the rainfall or the total precipitation. And we have a question for the feedback form later about maybe we should look at heat index or stickiness, as a recent paper called it, that combines the heat and the temperature, because that's most relevant to what it feels like from a human perspective. And then in the application that we're going to show, that's this data set is feeding the air temperature and the rain tabs. And then we're looking at sea level. NOAA Tides and Currents has this operational oceanographic products site where there's this API endpoint to extract the sea level data for the entire station history, and that's kept up to date. Here's a screenshot from their website of the data that gets pulled in, and that's the sea level tab, unsurprisingly. And then this last data set is for the NOAA coral temperature. And this is a 5-kilometer gridded data set. We wanted to look at the ocean, but there's also, we understand, in situ bay scale measurements that alternatively we could look at. This gives you more of a gridded approach of the sea surface. And you can see pretty dramatic changes from the historical values there under the ocean temperature tab. So these links are workable, so you can get more background information. And I put a link to the slides in the chat for those of you that on the chat in Zoom. So we're going to do a demo now. And just to get a lay of the land of some of these visualizations, we're just going to give these some terms so that when we get into the discussion, we can have a common reference. And so these are 4 different screenshots of different aspects of the app. And sorry, it's pretty small here, but it goes A, B, C, D. So left to right, top to bottom. And these I'm calling density cards, or excuse me, histogram cards, density histogram cards, as the overview. So that just gives this big picture idea of these values and how much they've changed from a historical climatological period compared to the most recent date. These swiping maps allow you to see gridded data and those pixel values to compare now versus then. And the trend plot is specific just to the sea level data, where we see a scatter plot of the values that vary over the years, and just a broad trend line over the entire time span of available data. So you can see that the rate at which the sea level is generally rising. And this daily plot, so on that x-axis is the day of the year, and then the idea is that you can see how the values change on the y-axis year by year, with an emphasis on the most recent the current year in red, and then the entire year before that in orange, and all of the years in gray. So that's not very intuitive until we actually jump into the app itself. And the link, this is just temporarily where I'm hosting it. There's a slide about next steps, which would be to operationalize this and put this on the TBAP server. But for now, it's living on my server. But all the code for this app is using our open source Shiny framework and on GitHub and publicly available. But because of this interactivity, there needs to be some server logic that updates what you're seeing as you tweak some of the configuration. So we're very interested in your impressions, and that's why we have a Google form linked in some subsequent slides. And so I just want to give you a very broad brush tour. I encourage you to also check this out. I'm gonna put the direct link to the app here, and so we have a navigation menu in the very top, and these are the broad indicators of interest. And this overview tab just really summarizes, okay, the most recent date, and sorry, this is about a month ago, just before we had our first child, so I haven't hit update on the data, but that'll happen. We wanted to get your feedback first about these indicators. And it's saying we have this broad value of difference between what is now in the red line and what was previously experienced in the white, the average of the years. And then that's what's called a density histogram, which is the frequency of values. So we do see that there's a high frequency of values for the air temperature that seems to correspond to the median. But when you take the average, it's the white line. That's just before that. And you can expand any of these by clicking on the lower right to get a closer view. And so we see that the air temperature has increased on average from when we compare it to the historical period. Rainfall seems to, on average, have decreased, but this is just for a given day. And ocean temperature is pretty dramatically of higher temperature. There's a lot of details that we can tweak with this to configure it. I mean, just for starters, probably degrees Fahrenheit would be much more accessible to most folks in the US, so we can do that. And millimeters could be to inches, we could do that. That's no brainer. And we also want to make sure that this can be visible on even a smartphone if people are accessing it that way. And this is going to look a little junky on your end, but I just want to demonstrate that, this is what it looks like on a smartphone, and the page lays out nicely to be able to view it that way with the cards. So that's the first brush. We wanted to have this overview. And then this air temperature. So Marcus and I had kind of scanned for other climate change indicator sites, and it just became a very compelling visual to have. Can you guys hear me?
Yes.
Yes.
Okay, cool. My internet just glitched, so I just want to make sure we're good. So this is defaulting to the most recent day, and I can swipe, and on the left I see the then period, which is May 22nd, 1981 to 2001, so a 20-year historical climatology, and versus now, which is May 22nd of this year. And the idea is to be able to, people can zoom in to an area of interest. We can see the Tampa Bay segments within that area. We can change the day of the year, and it will grab the average for that 20-year period, and compare it to now. We can go back to any day of the year, and if we go back to it would go back to 2023 if it's if we haven't yet reached that day of the year for this year, obviously. We can also configure this. So we didn't want to call that climatology, which is much more of a sciencey term. We want to say like then. Okay, that's is on board with that. And you can see what those values are. So it's this date and for that 20-year period, and it's the average on a per pixel basis. But we can also configure this, and you could be like, well, okay, that's just for one day. What about this average? So I can click this little slider and change that to, let's say, the most recent 10 years. And is it dramatically warmer now when we take the last 10 years and compare it to the first 20 years of this data set? And for this day and this comparison, you can see it's actually, yeah, it's actually significantly warmer, and it's visually strong to see that difference. Well, what about you basically can slide the years for the applying to then, and you can slide the years applying to now, and you can change the variable. And actually, this is just doing the average. This part doesn't work just yet. This is just a prototype to get your feedback, and we'll finalize all that. Okay, so that was like the most complicated visual, and with all the different options. We want to default to something that is sensible and understandable, but then make it also configurable to please any given interest. Oh no. Let's see if I go to a different bay segment. Let's see if we if that plot works for this one. Huh. Sorry about that. I am not sure. I can, yeah. What happened to that plot? So we had a map of air temperature and a plot that we were trying to look at, and I hit an error. It is the same graphic as this, but just the underlying data is different. So apologies for that little hiccup. But it's if we average all the pixels within a given bay segment, then we could see this. And so this air temperature, the ocean temperature is the same as air temperature, but it's using that NOAA Coral Temp data set instead of the PRISM data set. We see this slider here. And okay, weirdly, I'm not seeing the day of year option down below, but we can change this here. So it's the same. We're still working out a few bugs, but this is, we're seeing again this red is the current year, and on the x-axis is the day of the year. And so we're seeing this comparison of, okay, it looks like things are getting a little warmer. In fact, it's warmer than it's ever been. And then you could see other years past and the value for those years and the given date. So each line represents a different year, and 85 to 2023 are in gray. And we should probably add another thick line for the average of those past years, and maybe even a 20-year climatological comparison of 1985 to say 2005. But that's the idea, and you can also zoom in, and you can download an image, a plot as a PNG of any zoom level, and you can change the given segment. You can also look at this. Right now it's in dark mode. Excuse me, you can change it into light mode, and if you're printing something, that probably makes a little bit more sense. I think the dark mode just adds a lot more pop to the values of interest. So I started off with the air temperature, showed you the map. The plot didn't work, but it looks almost identical to this, except it's air temperature, not sea surface temperature. And then the rainfall is similar. The averages kind of smooth things out regionally, whereas for a given single day, you see it more spotty if there's a rain event, which makes sense. And we can also, yeah, just look at rainfall, which doesn't have this seasonal arc of summer being a strong signal. Like here, it's a little bit more chaotic throughout the year. But we have this Bay segment variation that we can look at, and we can zoom in, just like the day of year plot, because use the same Plotly tool to do that. Now, sea level is different. It doesn't have to be as different, and that's just something that we'd like to discuss with you is what this should look like. Right now, it's quite simple and just showing you the stations that are available within the Tampa Bay area, and then allowing you to look at individual stations and seeing the trend. So I can also similarly zoom in, and you can see that there's a rate of sea level rise. It's a little small, could be a little bigger for sure. And what the fit of that value is, which as that value approaches 1, it's a perfect fit. And as it approaches 0, it's more of a random fit. So I know that's a lot to throw at you all at once, but that's it's good to get this first brush. And if you find anything troubling, we especially want to hear from you. Or if you liked any particular aspect of this, we also want to hear from you. So just going back to these slides, again, here's the website, which I also put in the Zoom chat. We can talk about how the complexities of configuring any of these, how we could simplify these, how we can make them more detailed. And in this feedback form, so I'll just click to just so you can see it. It's a pretty simple feedback form. You can fill out all or none of this, and it's just hitting on these 3 questions of how are we doing with the indicators, the user interface, and the relevance to management, and any other comments you have, and also an optional email if you want to, if we could, follow up with you about any changes we made geared in that direction, or any clarifying questions. That'd be really helpful too. So with that, I want to go back to, indicators. How are we doing with these indicators? Did we capture the most important indicators? What are we missing? Some things that came to mind are, heat index. That's getting a lot of publicity these days because of the, there's like people are dropping like flies. No, sorry, very bad expression, but basically it's having a huge impact, and the temperature and humidity makes it more real to how it feels to folks, and also has ecological implications. And then what about water temperature changes? We use this station data, or excuse me, we use this satellite sea surface temperature data. Should we swap that out with in situ measurement? I'm just going to go over all these and then just open up the floor for broad discussion, and we can revisit these slides. And then should we be adding something about severe weather, extreme events? Obviously, hurricanes, that's not in there, but how do we make that accessible if we wanted to add it with intensity and frequency of hurricanes? And what's the area over which we would then capture historical data to come up with an indicator, a single number? And then drought, flood, weird events, hail, that we'd want to also capture? Are they happening more frequently and more intensely? The user interface. The user interface, is it intuitive? Is it clear what's happening? We're trying to avoid getting into modeled data that's projecting into the future and just capturing the change that is visible based on measurements made. The changing sea level, yeah, do we swap out that station map to actually show the values as we swipe of the then period versus the now period? Um, and then do we even have a similar daily plot? We take a historical period and compare sea level, so it's more of a stark difference, just for consistency's sake, or does it make sense to leave it as is? And are these configuration options, is it too much or not enough? Does it make sense to have that? And then on the management level, very broadly, are we are we um, are we providing something that has real management interest and is something that can be communicated and is consistent with um, existing TBEP activities with outreach and community engagement and whatnot. So just real quick, the next steps are we want to create more of a landing page, describe where this data comes from, limitations, caveats, assumptions, update the TBAP tools, our package with these new functions to update the data and operationalize this on the server. So you're seeing up-to-date data as soon as it becomes available. It's on the app and readily accessible. And this, just as a also a reminder, is just consistent with all the suite of tools that that Marcus has really been spearheading. And actually, I got involved with TVAP years back because of this paper that really outlines some of those concepts of just doing better science, more transparency, and more automated to make it accessible to all. So thank you so much for your attention, and please don't hesitate to reach out to myself or Marcus with your feedback, and we'd love to hear from you. Also, ideally with this form, so we can collate everything in one place and iterate and improve upon the app um, and serve Tampa Bay climate change indicator needs. So with that, I just want to hand it back to Marcus, and we can pull up slides and poke around the app, time permitting.
All right, thank you, Ben. I'm in the interest of time. Maybe we can move to the next speaker since we started late, but there's like an immediate question. I love it, I hate it. Whatever. Maybe time for 1 or 2 questions.
I have one comment just to keep you all abreast of things that are going on. For a number of years, I've participated on the Tampa Bay Climate Science Advisory Panel and started serving as a co-facilitator of that back in 2018. And at our last policy board meeting, at the request of many of your organizations as partners, they agreed that the Estuary program should take on the role of serving as the convener for the Tampa Bay Climate Science Advisory Panel. And so that sort of falls within our committee framework that Marcus showed that needs to get updated. So if you don't know that now, I'm letting you know. And the idea behind that Climate Science Advisory Panel is that we're serving the Regional Resilience Coalition, but we're also, serving you all in your individual capacities. And so our job is to break down science so that it's accessible and actionable for you all as practitioners. And so I think that these tools, in addition to coming before this group, need to go before the Climate Science Advisory Panel. But just wanted to let you all know that sort of organizational change has happened, and many of these indicators that Ben and Marcus have been developing are building upon the recommendations of that advisory panel that are included in the Regional Resiliency Action Plan that the Regional Resiliency Coalition has adopted. So just trying to put it into the whole regional context for everyone.
Marcus, a few questions which I'll
comments which I'll send to you, but real quick, one general is, I didn't see anything there about like data tracing where the viewer can like actually download the raw data and look at it themselves. Maybe it's in there, but I missed it.
All of the data driving the plots, maps is readily available. Well, it's available, I wouldn't say readily yet. But that was one of our goals, is to use data sources that were available from an API and update frequently. So that
I mean, it's like-
these updates. With our other indicators and dashboards, we often have like downloadable functions to actually get your hands on it, aside from going to the actual source and figuring it out on your own. If that's a feature that you think would be useful, we can certainly add that.
Yeah, and I'll just add to that all the data that you're seeing displayed is already on the GitHub repository with the code associated. So that's extracted specific to Tampa Bay. So that we can point to and have download links for very quickly. Yeah, good question. Thank you.
All right, I think we'll move on to the next presentation, but appreciate that, Ben. Kelly, what do we got?
Thank you.
Presentation from Amanda Morgan with USF on Carenia brevis and Pyridinium bottomense utilization of dissolved organic matter in urban stormwater runoff, rainfall entering Tampa Bay.
Share your screen.
Share the screen.
Thank you. All right, so thank you guys for having me. I'm going to be presenting some work from a project that was led by my advisor when I was getting my PhD at UF, Dr. Mary Lusk. And this project is named Up in the Air. I just want to thank TBER for funding this work and enabling me to do or kind of pinpoint these questions that I'm going to be going over for my PhD. And one of our collaborators was also Dr. Cynthia Heil, which she leads the Red Tide Institute at Moat Marine Lab. So there were several project objectives, but the ones that I really hammered into for my PhD were characterizing the nitrogen forms and trends in rainfall and urban stormwater runoff entering Tampa Bay. And I also wanted to conduct bioassays with Carenia brevis and Pyridinium bottomense to assess how organic nitrogen as well as dissolved organic matter, or DON, stimulates these local HABs, and that originating from runoff and rainfall. So getting into it, this is just some recent work that was published in the journal Water, Carenia brevis and Pyridinium bottomense utilization of dissolved organic matter in urban stormwater runoff and rainfall entering Tampa Bay. And as we all know, nitrogen is a pollutant of concern in Tampa Bay due to non-point sources, and it contributes over half of the N entering Tampa Bay from stormwater runoff. And atmospheric deposition is the runner-up at about a quarter of that total nitrogen load. And we're experiencing these recurrent HABs of Corynea brevis and pyridinium. And despite nitrogen reductions, chlorophyll A levels remain elevated in some segments of the bay. So could DOM, not just N, be part of the missing link here in terms of blooms? And why focus on DOM? Well, there are considerable amounts in stormwater and rainfall, yet little is known about the fate and reactivity of this material once it enters the coastal environment. And DOM is essentially just decay products of animal and plant material. It's also synthetic organics, so that could be urea-based fertilizers or glyphosate. And the technical definition is the organic fraction that passes through 0.45 micron filter. DOM is not just carbon, it's also reactive elements such as nitrogen, phosphorus, etc., and also trace metals. And a couple of terms I'll be using to define the origin of DOM: allochthonous, that which is externally delivered. That's an example of runoff, and autochthonous, which is internally produced. An example of that would be DOM produced by fire. And we're particularly interested in DON, or dissolved organic nitrogen, because anthropogenic activities have increased nitrogen export primarily in reduced forms. So that's ammonium nitrogen and DON. And one example of that would be the use of urea fertilizer. And we know that both of these species can utilize urea. And there has been a focus historically on inorganic nitrogen forms, ammonium, nitrate nitrogen. However, they make up a very small portion of the total dissolved and pool in our coastal waters. And DON is also dominant in stormwater runoff, up to 83% of total nitrogen, rainfall up to 57%. Briefly going over Corynea brevis, it's a single-celled dinoflagellate. It's a toxin producer. It produces brevitoxins, fatal to sea life, and it also produces risk to human health because these toxins can become aerosolized. And these blooms typically initiate in July and October months and then terminate in the spring. However, we have seen these blooms over summer. And they originate offshore where they're at background concentrations on the West Florida shelf. And upwelling dynamics brings them into coastal areas where they can take advantage of nutrient sources and coming from land-based sources. And for Pyridinium bahamense, it's also a single-cell dinoflagellate that produces saxitoxin, and this has resulted in more human illnesses and fatalities than any other HAB. And blooms are detrimental because they can impair seagrass because of light attenuation, reduction of water column oxygen, resulting in hypoxia-related fish kills. And they're unique and they form these resting cysts, which can overwinter and seeding new blooms. And they typically initiate in June upon warmer water temps in the bay and terminate in September, but we have been starting to see them prolonged. So why are these species so successful despite having a relatively low or slow growth rate at about 3 days it takes for cells to divide? They're mixotrophs, so they can not only take in inorganic forms of nutrients, they can supplement that carbon fix by photosynthesis using organic matter. So some of the ways they can do this: osmotrophy, phagotrophy, and they also have cell surface enzymes that can facilitate the uptake. And essentially they can outcompete strict photoautotrophic organisms when inorganic stores are depleted. And some of these sources of DOM are listed here that we know that they can use: humic acids, amino acids, soil extracts, urea I mentioned, but there's literally thousands of other DOM compounds in runoff. And I'm interested in seeing what these might be. And one of the ways we measure this is using fluorescent spectroscopy. So CDOM, chromophoric DOM, is that which absorbs UV divisible light. FDOM, or fluorescent DOM, is the fraction of CDOM that fluoresces. And these excitation emission wavelengths hint at the source of the DOM. And we can measure this using excitation emission matrix spectroscopy. It's really straightforward. You use a spectrophotometer and you don't need reagents, so it's a really nice method. And each EAM, since it's simultaneously scanning at different wavelengths, as you can see in this figure on the left, gives you kind of this elephant under the blanket. And to piece apart these individual fluorophores, we can use parafact modeling. which essentially is a form of mathematical chromatography. And each of these underlying spectra can tell us if it's humic-like, soil fulvics, protein-like, etc. So it's really useful. So getting into the sampling regime for this experiment, we wanted to sample runoff from bloom relevant sites. So for Pyridinium bahamense, at the top, you can see those were outfalls that were sampled in Safety Harbor and Clearwater. So that outfall is directly emitted into the bay. And for Karenia brevis, we had 2 sites that were collected. One in Treasure Island, which is on the beach side. It's a Finger Island development. And that was collected as water runoff went into a curb inlet and exited directly into Boca Ciega Bay. And also we sampled a mainland runoff source from this outfall that you can see in the middle. And that was during low tide to prevent any contribution from Boca Ciega Bay. Atmospheric deposition was collected in an open lot devoid of tree cover in Pinellas County. And this was done in June for Pyridinium bahamense and July for Karenia brevis. And the experimental design was identical for both, but they were done at different times. So we used the Moat, Minnesota clone for Karenia brevis. And for Pyridinium bahamense, we had isolates from FWRI, the Indian River Lagoon strain, and both of these are non-xenic cultures. And we wanted to really probe into how different intensities of rainfall could potentially trigger blooms. So we tested or simulated rather a low rainfall event, so 0.4 inches, and also a high at 2 inches. And then those 2 stormwater sites for each species. This lasted for up to a week. We measured cell concentrations, nutrients, so inorganic and organic nitrogen species, and also dissolved organic carbon. And then we characterized that DOM. And in doing that parapack analysis, we were able to define 3 components of DOM. C1, which is a terrestrial humic-like component. C2 is a microbial humic-like component. So essentially that's humic-like DOM that's more freshly made by microbes. And then C3, which is a protein-like component. And in general, we saw depletions from the terrestrial-like component, which is that salmon color in stormwater for both species. So the stormwater is on the far right of each one. So beachside and mainland on the left for K. brevis, and then on the right for P. bahamense, Clearwater, and Safety Harbor. And interestingly enough, when we added these treatments, we saw stimulated production of that humic-like DOM and protein-like DOM. And that's important because this material can be recycled and lead to bloom maintenance on longer time scales, as this DOM is considered highly labile. Both of these figures are showing the nutrient nitrogen concentrations in DOC. So Karenia brevis is on the left and Pyridinium bahamense is on the right. What we found was when we calculated bioavailable DON, we had up to 75% for Karenia brevis within 72 hours and up to 100% for Pyridinium in 48 hours. And then following that, there was a preference for ammonium nitrogen instead of nitrate nitrogen, which is unusual for Pyridinium bahamense. Typically it's the opposite. And there was also a bioavailable DOC that we calculated for both species. And for Karenia brevis, it was greatest for the beachside runoff treatment at 28% and lowest for the mainland treatment, which could mean that those inputs were more recalcitrant. In terms of BDOC for Pyridinium bahamense, we saw the greatest for that high simulated rainfall event in both runoff treatments, not so much for the low. This is the growth response, so cell concentrations in cells per liter for both species. Karenia brevis is on the left again, Pyridinium bahamense on the right. And the magenta and the blue lines are both runoff treatments for each one. And we saw significant increases in cell yield, so maximum cell concentrations and also specific growth rate for both species. In terms of the rain high simulated treatment, we saw significant increases in specific growth rate for Pyridinium bahamense only. And that low treatment really corresponded well with the controls. So controls are that orange color, salmon color, and rain low is that lime green color. So this suggests that maybe these less intense rainfall events might not be as problematic as these more intense higher rainfall events for these blooms. Of course, we need to do this in the field, which is ongoing, but I'm going to get into some field monitoring now of runoff and rainfall that we did in Tampa Bay over 2 wet and 2 dry seasons. And our goal for this was to quantify and characterize the seasonal spatial inputs of DOM again and nitrogen entering Tampa Bay via rainfall and runoff. And we really wanted to put this in context with blooms of Carenia brevis and Pyridinium bahametis. So we were able to calculate the monthly fluxes of nitrogen and DOC from each site. We wanted a monthly time step instead of annual to see when the greatest fluxes into the bay would be. And we characterized the DOM again, comparing wet and dry seasons. We also did some isotope work with nitrate isotopes, stable isotopes, and again, put these in context with HABs. The sampling and methods were the same as the previous design. So rainfall starts, you go grab some samples. We had a site in Gulfport, which is the top, which is that great inlet structure. The one right below that is the Lassing Park outfall. And then we had 2 curb inlets that we sampled in Treasure Island again, and then one was in Old Tampa Bay. And rainfall again was an open lot and over 2 wet and dry seasons, and this began in July of 2021 throughout February of 2023. And all nitrogen species, again, DOC, EM spectroscopy. And this is how we estimated the monthly flux. So we delineated a subcatchment for each of our sites, and we used the stormwater network. So we looked at continuity in the stormwater conveyances to see how much of that area was contributing runoff to where we were sampling. So for example, how much of that subwatershed was going into that curb inlet. We also used LiDAR to assist with this. And then we used a series of equations that you can see here that I can get into if you would like. And then rainfall, same thing. And we tried to estimate annual TN loads to compare them to current estimates as well. So going over the nitrogen concentrations, what this figure is showing is every single sampling event for each nitrogen species and each corresponding site. And the dates are repeated on the x-axis. So in general, DON was the dominant nitrogen form in stormwater across all sites and all seasons, except for Treasure Island, which was delivering predominantly nitrate. And DON was significantly greater at Lassing Park, Outfall, and also Gulfport during the dry season. In terms of the dissolved inorganic nitrogen species, nitrate was the dominant form for both seasons, except for, again, Lassing Park and Gulfport. So these sites are really delivering reduced forms of nitrogen. And for rainfall, DON was up to 55% of total nitrogen. Again, it's dominating the end pool, and nitrate was dominant in the dry season, and that switched during the wet season. And some of this isotopic work, this was done using a Bayesian mixing model. And the sources we were interested in are atmospheric, ammonium, and nitrate-based fertilizers, sewage and manure sources, and soil and organic nitrogen. And corresponding to some of the previous work by Dr. Lusk, we saw atmospheric nitrate nitrogen contributing the most, except for Lassing Park, where we were seeing more sewage and manure inputs. And then that was the second highest contributor for all other sites and seasons. And we saw contributions of nitrate, nitrogen, fertilizer that were higher than ammonium. And this was across all sites, and this was inclusive of the wet season. And going over the dissolved organic matter characterization, we compared the wet and dry season again. So this is just showing percent contribution. The wet is on the left, the dry is on the right. And the components that we were able to measure, again, we had that microbial humic component. We now detected a soil fulvic component, terrestrial humic inputs, and also a protein or tyrosine lake component. And what we saw were distinct shifts in the composition from the wet to the dry season. So we had more labile fractions, so the microbial humic and the protein DOM during the wet season. And shifting to the dry, we only saw inputs of soil fulvic and terrestrial humic. And looking at rainfall DOM, up to 79% was protein-like in the wet season. And what this tells us is that the wet season is really a critical time period for the delivery of labile DOM. And this is a lot, but I'm gonna briefly kind of go over this. These are just showing our monthly fluxes of DOC and nitrogen. So far left, we're looking at the nitrogen flux for the entire sampling interval from July of 2021 all the way to February 2023. And DON is in the red, ammonium is in the blue, and the nitrate is in the green. And then going to the middle, we have same figure, but now we're just looking at DOC. And then all the way to the right, we're looking at the monthly rainfall. So what we're seeing essentially are the fluxes of DON and DOC were the greatest during the wet season, except for Lansing Park. That was always an outlier in this study, namely in the months of August and September. And an interesting finding from the study was that rainfall DON flux was 14 times greater in September of 2021 versus 2022. And if you all remember, we had a horrible Piney Point wastewater spill. So we're hypothesizing that this was due to biogenic emissions of DON because we had higher primary productivity in the bay at that time. And this is really interesting because this is essentially showing that whatever we input into the system can be released biogenically and potentially lead to atmospheric fertilization once you get another rainfall event. And that has implications for bloom maintenance as well. And we estimated the annual TN load, and this is wet and dry deposition for 2022. We used 2022 because that was the only year we had a complete sampling interval. And we estimated that at 1268 tons per year versus the most recent on the TBEP websites, 435 tons per year. We're thinking the discrepancy could be because our site was in the urban watershed of St. Pete. And my understanding is that the Verna Wellfield site in Sarasota is used for the TN concentrations for current estimates. So that could show maybe some localized monitoring sites could kind of minimize those discrepancies, but that remains to be seen. And in terms of DOC loads from rainfall is almost double that of total nitrogen. So we're getting considerable amounts of carbon and nitrogen from rainfall. And we also found that rainfall depth was a significant driver of both DON and DOC export. So that has implications with climate change. If we're getting more rainfall intensity, we can hypothesize that some of that material will be exported. So just to wrap everything up, the results from each bioassay show that stormwater inputs and rainfall support both species by providing humic substances, but also DON, DOC, and ammonium subsidies. And rainfall and runoff provide stormwater and nutrients that vary by site and by season. And putting fluxes of these sources into a monthly context, as we did here in future years, may help reveal drivers of water quality impairment in Tampa Bay. And I want to thank you all for having me and supporting this research, and any questions are welcomed if we have time.
We have questions? Have you done any accounting for the application or over application of reclaimed water in Lansing Park directly adjacent to the bay? Because I assume there's no attenuation.
No, but repeatedly at Lansing Park and actually in Treasure Island, there was application of reclaimed water while I was sampling during rain events. And I think that's contributing to the wastewater signal that we're seeing there potentially. But that just goes to show that that's something that management wise, maybe we can work on in
And
terms of not.
that's high nitrogen wastewater
Yes,
too.
yeah,
So,
absolutely.
thank you.
Sure.
I'll also just provide another contextualizing comment. This reestablishing localized atmospheric deposition monitoring stations is an action that's been in the CCMP. It's a priority, and we haven't really made any progress on it since the early 2000s when we did the BRACE study. It's something that's come up repeatedly at the Nitrogen Management Consortium. And I think we'll likely be making a push to get a task force together of folks on the air quality side, the monitoring side, to see if we can advance that particular action, because we recognize it's probably been an action that's been deferred too long. And if we really want to understand these nutrient sources, we're going to have a site closer than just Verna.
Anybody else? Online?
We've got one in the room.
Carrie. Great.
I just have a question about your bioassays, and we can talk more. I know we're going to talk after this, but I was wondering with the pyridinium, it looked like your starting cell concentrations were pretty low, and you had some carryover from the culture, possibly because your inorganic nitrogen was relatively high in your T0. And I was just wondering, how, like if the growth that was supported in those first few days could have been supported by that carryover from the culture, which was just
Absolutely.
what you thought about that.
Yeah, and actually I had grown them in instant ocean to try to deplete any extra organic to really focus on the change in the DOM. But essentially whatever is in culture that's left over, and if I'm not adding more nutrients, they can utilize what's there, especially on cell lysis, which I'm sure you know. But I think the important part of that, even though there was a drawdown in inorganic, we saw the DON was important too. So I was just trying to focus on the organic fractions, particularly in the additions. Yes, there was still ambient inorganic left, at T0 that we were able to track that, but I think it was really just the change in those species that we were trying to focus on.
Yeah, it suggests there could be other components that would be beneficial in that, what you mentioned in the organic matter,
Sure.
that other than nitrogen. That
Sure, and I think one of the interesting things too from the bioassay is when I sampled, I wasn't able to detect any protein-like DOM in the runoff that I added. So that's another thing, like when we have this wet season protein-like DOM, what does that look like in terms of pyridinium utilization too? So that's just something that I wanted to look into, because typically amino acids are pretty bioavailable. And we saw a lot of it in rainfall alone. So, but yeah, something we can talk about for sure.
So just based on what you said, I had a follow-up. Like you were looking at the drawdown of different forms of nitrogen in the culture flasks. Just looking at the drawdown, it's difficult to say if the growth was actually being supported on that.
Sure.
So even if they were utilizing, they were not, even if DON was going down, we don't know if they were utilizing DON because dinoflagellates are well known to form these pools where they store nutrients
Yes.
and different forms of nutrients. So the fact that some of your experiments or treatments started at a pretty high concentration of DIN too, a lot of growth that you saw may have been on DIN.
Sure, but I think an important part of that too is even with DIN subsidies, DON was depleted. So it's showing that there's
That
not.
they are taking it up, but not necessarily growing on
Yes.
them. Because you would have to do isotope experiments to really prove that they were
Sure,
growing on those sorts.
To be able to say definitively, this is what they were using. But when you're tracking the depletion in terms of the growth concomitantly, we can just kind of make these inferences that they were utilizing DON as a nutrient source.
If you had no other source of
Yes.
nitrogen.
Absolutely.
But your treatments did have other forms of nitrogen.
Yes. So are you asking me if I were to do this again, I should isolate it so that they're only getting DON and not any inorganic nitrogen to isolate that they're using the DON only?
I guess I'm saying that when you are interpreting your results, that's a very important part to consider, that you were starting with really high concentrations of DIN,
Right.
and the growth that you were seeing may have been just driven by DIN, and the fact that DON came down
Right.
doesn't, like, it's not a cause and effect relationship there, because there was more than enough DIN for them to keep growing.
Sure. So then I guess my question to you would be, why would that DON be depleted?
Because in general, a lot of phytoplankton, dinoflagellates are known to do luxury uptake of nutrients if they are available, because usually in nature, dinoflagellates always come in nutrient depleted waters. Spring bloom will happen, which is dominated by diatoms, and then dinoflagellates come after. So they are evolved to do luxury uptake of nutrients, which they will do in whatever form.
Sure.
But the fact that it is more of an energy to break it down and make it in a more usable form for them,
Sure.
they may just keep it for those, it's like their bank account for emergency.
Sure.
So they may have taken it up, but not necessarily utilized to grow.
Sure.
And the fact that your experiments did start with pretty high DIN concentrations, it's very difficult to tease it apart and infer or assume that the growth that you saw was supported on DON.
Sure. And I think you're making a really good point in terms of luxury uptake, because if we're adding more organic nitrogen to the system, that can be taken up. And then when those inorganic nitrogen sources are depleted, and this utilization of those organic nitrogen compounds for growth.
They may, but like, yeah, it will take a lot of experiments to prove that.
Okay.
They may, because they produce toxins also, which are nitrogen rich toxins.
Yes.
So they may not utilize those nitrogens necessarily for growth, but they may just biotransform it into other forms.
Yes.
So like, your results do show that they can take up DON, but what do they do after that
Sure.
is still a question mark,
Sure.
is my point.
And I think this is something that I would love to talk to you about too, in terms of natural field populations, because as we know, what we do with cultures can go one way, and then when we actually do this, in old Tampa Bay. So, I mean, I would love to talk to you more about that, and great experimental design at some point to see what's going on.
Yeah, no, absolutely. We would like to talk more about the work you did.
Thank you so much.
There's a question online, Natasha, if you want to unmute.
Yeah, hi. I'm just on the more practical side. This is fascinating, and I always love hearing what you're coming up with in your lab and your work. But I'm just thinking about it from a monitoring perspective, and how feasible at this point with some of the other stuff we've been talking about. Do you see monitoring these stormwater, and I'm just saying blanket parameters because there's a lot of different species you talked about. And how long does it usually take to get from like a data collection to a result for any of these parameters? And how much would it cost at this point? Is this something that a lot of people are doing, or is this kind of the onset of some of these analyses?
Sure. So at USF right now, I'm kind of spearheading monitoring DOM at our T-bond sites. I don't know if anyone's familiar with that. It's the Tampa Bay Observing Network. We have 5 sites in lower Tampa Bay that we have exosons doing continuous measurements of water quality. On a monthly time step, we're also collecting nutrients in DOM. So that's my goal, is to kind of keep the change in DOM within the water column in context with more high resolution sampling with the in situ monitoring. And yeah, hopefully expand that into Old Tampa Bay, because we know we're having water quality issues there. But yeah, that's ongoing, Natasha.
Thank you.
Thank you.
Do you have any additional questions? All right, hearing none. Thank you very much, Amanda. That's Jerry with Stantec, the Old Tampa Bay Assimilated Capacity Project. I'm sorry if I did not get your last name correct. I gave it a shot.
Just kind of roll with it at this point. And I should just introduce real quickly, beside what I'm presenting here, Miles Medina, who's also part of our team, is gonna be talking about some of the work that we're currently working on, and I'm gonna kind of give a summary of where we've been. Okay, so this is an update on the Old Tampa Bay Assimilative Capacity Assessment Study. Basically just an overall project overview, and then as you're pretty familiar with the status of Old Tampa Bay, just kind of a reminder of why this project kind of came into being in the first place. So looking at the various bay segments, there are some challenges that were noted within Old Tampa Bay. We've kind of covered it already and we're all aware. There's nutrient loading concerns. In general, there's some poor circulation because of all the causeways. And as a result, there's been, in recent years, non-attainment of the chlorophyll A targets. That has led to, while there's been general recovery in seagrass population coverage within Tampa Bay, there's been some alternate trends and notices of seagrass losses within Old Tampa Bay. And as we've just heard from a couple individuals, but there's been some issues with pyridinium blooms. This is looking at the regulatory threshold for Old Tampa Bay. It's an annual mean of 9.3 micrograms per liter for chlorophyll. And then the management target is actually the 8.5 micrograms per liter. So Marcus and others have shown this graphic, and it's on the website, but it's just showing that it's alternated between attaining and not attaining that target as time has gone on. Even though in the last couple of years, there's been lower chlorophyll A concentrations in general, in Old Tampa Bay as a whole, there's been higher concentration observed over time. This has also led to, even though there was that lower chlorophyll A concentrations noted within the last couple of years, there's also been a seagrass loss noted. More specifically in this portion of Tampa Bay is the haliduli. So that on the graphic is the second line, like the orange, that shows that a drop off, and that has led to the overall seagrass decrease in the last couple of years. So that's why this project came into being, is you've seen some success in Tampa Bay as a whole, but Old Tampa Bay kind of has something else going on. So the 4 tasks that were required for this project were task 1, to evaluate the existing management paradigm, which I'm sure you're all familiar with since a lot of work went into it. I'm not going to show the complicated model of all the inputs and drivers and impacts, but it is a very involved paradigm. So that task is actually complete. The next tasks that are kind of concurrently being worked on right now is task 2, which is evaluating the eutrophication targets, threshold, and nutrient criteria. Task 3, the management intervention assessment, and task 4, which is reevaluating existing nitrogen allocations in Old Tampa Bay. This was kind of a calendar year where the various tasks and deliverables and some meeting dates on here. And this is about where we are. So like I said, task 1 is complete. That document is finalized. It's online. I'll provide a link to it next in the presentation. And it's also on the Estuary program's website. We're currently in the throes of task 2, which is evaluate the eutrophication indicator targets. We have a draft deliverable, and a final document coming up in the end of quarter 3. And then concurrently working on tasks 3 and 4. So we're looking at the targets, and then ultimately, how does that reflect to nitrogen loading? And does that need to be reevaluated? So first of all, we're going to go into task 1, what was already complete. So like I mentioned, there was a very comprehensive paradigm, and that was showing the ecological interactions as well as the nutrient management portions. And it basically emphasizes nitrogen loads as the primary lever. So what can we control? What can we regulate? So the idea was that if you reduce the nitrogen loads, next step is that you would reduce the chlorophyll A concentrations, which in turn would increase the water clarity and increase the seagrass coverage. As part of that whole effort, there was a very comprehensive model that was used that was supported by the integrated model. And that took into account multiple different submodels, if you will. You have the hydrodynamic model, which is the ECOMSED model that is looking at winds, humidity, et cetera. You have the watershed model, which is utilizing the rainfall and land use. You have the water quality model, and then you have the optical and ecological model. So as part of our effort, we reevaluated what was done. We looked at these models, what was input, what was output, and had some, critique on what could potentially be improved, what was missing to give a better picture of what is actually going on in Old Tampa Bay. So was a completely new paradigm needed? So given the uniqueness of Old Tampa Bay watershed and the expression of nutrients, how do we better manage? We know there's some changes hydrology. There's general poor circulation to that part of the bay compared to other regions or other subregions in Tampa Bay in general. We've talked about and seen the increasing temperatures over time. And we've just heard about the increasing pyridinium blooms. So how do all these things interact with each other? And how do we help potentially solve the problem that is in Old Tampa Bay? So what we found is that it's managing for total nitrogen loads and taking into consideration the other drivers are what's needed. So it's not just solely looking at the nitrogen inputs, but what are the things that we can't control, like temperature, right now flow, and hydrodynamic exchange between water within Old Tampa Bay. And that's what's all needed to try to improve the overall seagrass coverage. So the other thing that was discussed in the report was that Old Tampa Bay should be looked at more of a gray box instead of, we know all the interactions and how everything works together. And it's only partially understood. So we basically are trying to manage a watershed under uncertainty and complexity. So for this evaluation, we looked at what management levers can we tweak. So like I said, can we look at nitrogen loads? Do those need to be altered? Do the timing need to be altered? The freshwater inputs, could there be some controls of when those occur? And with circulation, can there be more openings in Tampa Bay? How do we make that better? And those all have to accommodate for the stressors that are out of our control. So temperature, sea level rise, and extreme weather events where you have pockets of high rain in input versus an overall average rainfall. And basically the whole idea is that all these different levers would potentially be tweaked. And instead of controlling for water clarity, which was what we thought was what can be solved, it's more of, we need to reduce the algal blooms and especially pyridinium, because the pyridinium is the thing that is having the impact on water clarity and consequently, as far as we can tell, having an impact on the seagrass recovery. So like I said, all of that information in way more detail and evaluation can be found in this report. This is a screenshot of the document online, and then this is a link to that deliverable, which is on the Estuary Programs Library. So I'm going to turn it over to Miles to talk about some of the path forward and where are we at currently and for future evaluation for tasks 2 through 4. So Miles, let's see if we can hear you.
All right. Yeah, can you hear me okay?
Yep.
All right, awesome. So yeah, in this section, we want to just give a sort of high level flavor of some of the approaches we're taking. Not so much talking about specific results. This is still ongoing work, but just give an idea of some of the approaches and methods that we're using so far. So one of the things that came out of task 1 was the realization that there's this discrepancy between the actual nitrogen loading coming off of the Old Tampa Bay watershed, which is shown in the top panel there in blue, and the normalized, hydrologically normalized loads, which are adjusted for based on runoff and ultimately rainfall during the early 1990s reference period, 92 to 94. So the discrepancy is basically that if you look at the threshold, which is that dotted line, the regulatory loading rate threshold, if you normalize the loads for hydrology, so basically if it rains more and you get more load and you make an adjustment for that, you effectively discount the load. Then we've things have sort of been in compliance for a couple of decades. But of course, Old Tampa Bay is dealing with the actual load, and of course with additional rainfall and runoff, you get some amount of flushing and dilution. But the actual loads are tell quite a different story. So maybe the most stark uh, discrepancy is 2015. There was about twice as much rainfall 2015 compared to the reference period, as shown on the bottom plot. And you can see in the top plot how the difference between the actual and normalized load is that factor of about 2, right? So it goes from something like 400 tons that year to something over 800. Now, without getting into too much detail, we do have, like, based on other analyses, we do have some indication that achieving the current load target in terms of actual loads might be sufficient to see affect the changes that we want to see in chlorophyll and paradigm blooms, without having to lower the actual threshold. It's just if we count in terms of the actual load versus as opposed to the normalized load, that might be enough to get us there in terms of chlorophyll. Okay, let's see the next slide. Okay, now in terms of attaining the chlorophyll threshold, one thing we've been working up, and this picture on the right is just a sort of demonstration example. So it doesn't really convey any meaningful results. But what we've done here is construct, basically it's a logistic regression where the output is the probability of attaining some threshold. So in this case, we're using the regulatory threshold of 9.3 micrograms per liter of chlorophyll. And the idea is that we can we can build this model up considering total nitrogen load along with other things like temperature, salinity, and so on, and derive estimate a probability of being below the specified threshold, in this case 9.3. And what these two curves represent, or could represent, for example, is maybe the blue curve and band are under a higher temperature condition, for instance. You'd have a lower probability of attaining the threshold for a given nitrogen load, as opposed to the pink band, which would be a lower temperature sort of condition. So this allows us to kind of play with scenarios using history as a guide, based on observed data over the last couple of decades. And again, we can we've built this model so far just using like one variable at a time. So in addition to nitrogen load and chlorophyll, or probability of chlorophyll on the axes here, sort of behind the scenes, we might have temperature or salinity and so on. But ultimately, we would be working with a model that includes suite of these variables. And of course, we'd also want to account for nitrogen loading with some delay, like 2 or 3 months of delay, not just nitrogen loading during the same month as the chlorophyll observations. All right, next slide. So this picture gives us an old Tampa Bay wide view of what's going on with Pyridinium during the period of record. This is based on data from FWC. They do typically biweekly sampling throughout Old Tampa Bay during blooms, which occur very regularly. They start around April or so, and they can last till later in the year. So the sample data are shown in the top panel. I've actually done some signal processing on these data, and it's extremely regular and seasonal. It's really quite remarkable to see such a strong signal in biological data like this. The bottom panel on the left, panel B, just shows the sort of descriptive statistics for these blooms on an annual basis. We've excluded the data that have no presence of pyridinium. So samples with a 0 cell count aren't included here. So you can see how the median and the middle 50% of the data in the minimum and maximum range have changed over time. It's been somewhat of an increase overall since 2018, with the last couple of years in the record showing a little bit of a decrease. Likewise, in the next panel over in the middle, you see the period of time each year when a bloom occurred according to different thresholds. So we have low, medium, and high. That's basically orders of magnitude. So going from low at 10,000 cells per liter, medium is 100,000 cells, and high is 1,000,000 cells. And so you see, when those blooms have occurred, and get some impression of the duration of low, medium, and high conditions each year. Notably, since 2018, you see that thicker sort of red band in the middle of the bars has increased. So that's a sort of longer duration conditions with over 1,000,000 cells per liter. One thing to consider here is this is a very simplistic sort of definition of the beginning and end of a bloom. Generally, this definition works, but what we've done is just taken the first and last date of the date of the first and last sample each year that exceeded each of these three thresholds: low, medium, and high. That generally works, except if you look at the plot above in 2012 and 2023, there's this sort of gap where you could reasonably consider that like two separate blooms. So we kind of gloss over that gap and consider the entire thing. So it's a little bit simplistic, but it still gives an idea that blooms seem to be overall increasing in duration to some extent and intensity since 2018. And then finally in panel D, we see the duration of bloom conditions at each threshold, low, medium, and high, each year. So again, since 2018, we've generally seen longer lasting blooms. And and one final note on panel C there on the bottom sensor, you see that the blooms seem to be starting earlier each year. So that partially explains the longer duration. That probably has something to do with temperature, increasing temperature. All right. So, excuse me. The way we've been doing these analyses is generally looking at Old Tampa Bay as a whole, and we're mostly relying on water quality data from EPC monitoring, which goes back at least a couple decades. And then we're also looking at conditions within each of these six subsegments here. So we have sort of a west and east side of the north, central, and southern parts of Old Tampa Bay. The boundary between the north and central is the Courtney Campbell Causeway. And then we have the Howard Franklin Bridge between the central and southern segments. And the southern segments straddle everything south of the Franklin, straddling Gandy as well. Yeah, let's look at the next slide. All right, so this takes a little explanation, so I want to walk through how to interpret this graph. This is something kind of new. We wanted to have an idea of, whether reducing the chlorophyll target would be like an effective way of trying to control pyridinium blooms. The first thing you might think to do is just take, paired data, let's say the monthly average chlorophyll concentration and the monthly maximum pyridinium cell count, and put them up in a scatter plot and do a regression. And you can do that, and in some cases, you'll see a significant relationship there. You'll see a correlation. I think it maxes out at about 0.45 in one of the subsegments. However, within the relevant chlorophyll range, like let's say 0 to 10 micrograms per liter, there's still a lot of variability in the pyridinium, maximum pyridinium abundance. So you might see, you know, sort of the minimum pyridinium cell count, you know, about 300 cells per liter, all the way up to like 100,000 cells, which is considered a medium bloom. And so there's not really a useful relationship there that can tell us, you know, do we need to adjust the management target or the chlorophyll threshold down to better control pyridinium. So what we did instead was think of the chlorophyll threshold as you know, as it is a sort of maximum, right? I mean, this is like when we set a threshold or a target, we're saying we don't want conditions to exceed this level, chlorophyll concentrations to exceed this level. So by way of example, if we look at the top panel there, what this is showing is during the period of record 2012 through 2023, this is showing monthly maximum pyridinium cell counts that occurred during months when the monthly mean chlorophyll concentration was at or below, in this case, 9.3. So if you take all the months where your chlorophyll was, you know, below the current regulatory threshold of 9.3, this is what this is the distribution of monthly maximum values that were observed for pyridinium. So now we have a distribution. We can characterize it using the center and the spread. So the center is that vertical blue line that's a little bit above 10 to the 4. So it's the median value of this distribution, and then the middle 50% of the data are contained within those vertical blue dotted lines at around 10 to the 3 to 10 to the 5. So that gives us an idea of the spread of the distribution. So that's the pyridinium maximum, you know, distribution associated with this particular value, 9.3 micrograms per liter. And the plot below, what we've done is basically look at how that distribution changes as we change the chlorophyll value on the horizontal axis. So the red vertical line in that bottom graph corresponds to the distribution that we see above. You see that median at about 10 to the 4, and then that middle blue band is the interquartile range, the middle 50% of the data, which as we see above is about 10 to the 3 to 10 to the 5. And what you want to look for in this plot is where is there like a big step down as you decrease chlorophyll, right? I mean, it's possible that changing chlorophyll doesn't, you know, the chlorophyll value on the horizontal axis doesn't really change the distribution of maximum pyridinium cell counts. And in that case, you would just see sort of a flat line all the way across. But what we see here is as we move to the left from 9.3, it's relatively flat for a while. But once you get past 7 micrograms per liter and down to about 6, then you see this really steep drop off, which suggests, you know, at least historically, that the pyridinium values associated with this lower chlorophyll level attainment of a lower chlorophyll threshold would be substantially lower. So this is just a sort of, you know, course overview or you know, first look at what neighborhood we might be talking about for reducing the chlorophyll target. A couple of things to note here are that, of course, here we're looking at Old Tampa Bay as a whole, and there are really important discrepancies in different subsegments. So we've actually done this on the subsegment scale as well. We have a pocket slide showing those results if there's interest during the Q&A. The bottom line is that the Northwest and Central West subsegments, so that's like Safety Harbor, Feather Sound kind of areas, those are really driving what we see here in this plot. And then the other thing is we're looking here at monthly data when, of course, the target and the threshold are for chlorophyll are expressed as annual averages. So translating from monthly to annual, you know, is something that, you know, how we do that exactly needs to be decided. For example, you know, would we want a management target that for the annual average, but also like a wet season average, you know, and maybe 2 different targets since these blooms are so seasonal. And then we might also want to incorporate, we can do this with different variables. So we've built this up with a lot of different things, not just chlorophyll, but also nitrogen loading, for example. And there we can incorporate some delays and so on in nitrogen loads. So again, this is just sort of a, like using history as a guide, what have we observed in the past in terms of average chlorophyll values on a monthly basis and the associated pyridinium levels. And this can be a basis like that we can refine further to help inform a new target or threshold for chlorophyll.
So speaking of that, besides some of what Miles just talked about, he's also looking at doing some empirical modeling. And then the other thought that we had was to look at potentially a different model, which is the chasm model. So the first couple models on here are those that have already been used, as well as the updated Tampa Bay box model. But Steve Bartel, who's on our team, part of his background is looking at watershed ecological models, and so there's the chasm model. What makes this one unique is that it looks at multiple variables and multiple biological variables. So because there are different types of algae and phytoplankton, we talked about diatoms and dinoflagellates, it can parse those out and look at a finer scale and look at more potential changes over time. And it also incorporates things that were not included on the water quality side in the previous model, which was salinity. So we can look at those various water quality parameters, how they're interacting with the biology to provide some more weight of evidence of what is the path forward. Some of the suggested numbers that Miles had on there, that decline between 7 and 6 micrograms per liter, is that looking like historically with modeling? Does that how it parses out? And then moving forward, how are with the anticipated changes in temperature and potential salinity pulses, like how does that impact the ecosystem? So this is kind of our next steps of looking at exploratory modeling with a different method that takes into account other variables and factors on the ecological level, which is what seemed to be somewhat missing from the previous modeling and analysis efforts. So that leads us to our path forward and how can the TAC be involved? There's kind of two options. There's the do nothing option, which I don't think anybody wants. With the do nothing option, you're gonna have continued eutrophication. You're gonna have the persistent pyridinium blooms. How bad could they get? That's a question out there, as well as how much more seagrass loss is possible and is that going to expand? And you would have the failure to meet the numeric nutrient criteria and the various targets and thresholds for Old Tampa Bay. However, for option 2 is to revise the current management plan, and that could be either targets and or thresholds. This group is more interested on the target side of things. So some of the things that we presented in the steps forward that Miles first talked about was looking at the system for total nitrogen loads, not hydrologically normalized, and looking at those numbers and how they have a direct impact instead of kind of silencing the variability that you see. Potentially looking at reducing the chlorophyll A thresholds, is that bay-wide or targeting some of those sub-segments, that northwest quadrant, central west, is that feasible to have that because of the interaction that exists currently hydrologically? Would potentially down the path of reducing nitrogen inputs in specific areas. Previously, we just heard about the different nitrogen inputs. Could that be, looking at changing what type of forms of nitrogen are added to Tampa Bay? We won't bring up the terrible word. I won't say the D word in here, but there's removing legacy nitrogen within Old Tampa Bay and potentially removing additional barriers to hydrologic flow. So the questions for you all is focusing on chlorophyll A and algal blooms as a management endpoint. So where we would need some help and weigh in on decisions forward is what are potential changes to those chlorophyll A management targets? Should there be changes? And if there are changes, what should they be? Should they be for all of Old Tampa Bay are targeting Libid because there's 3 water body IDs kind of separating the north, central and south. Does it need to look at that area or does it need to be a smaller scale of subsegments? And then the second part was spatially targeted interventions informed by spatially targeted analyses. So looking at the nitrogen load sources, discharges, and review and provide input on revised, potentially revised nitrogen loading targets. And these targets could be on a spatial, temporal, like Miles said, like, do we have to limit what's coming in the wet season versus dry season, or the source? Is it non-point runoff atmospheric, which that has the least control, but you know, how do we help the problem that exists? And that is all we have for you guys. And this is just a link to the deliverable once again. I don't go back to the questions for you to ponder on. I have questions for myself and Miles. If there are.
Is this coalescing for people?
There's a question online from Chris.
Go ahead, Chris.
Yeah, hey, everybody. Yeah, good stuff. I did have a couple of thoughts I just wanted to kind of share, but it kind of speaks to at least the first bullet here, which is the idea of chlorophyll A as an endpoint. I really like that figure, Miles, that you showed that had that sort of that increase in pyridinium versus chlorophyll A, that one figure on the bottom right of that. Yeah, that one right there. But the thing I guess that I'm struggling with a little bit with this is, you know, chlorophyll A is simply an expression of phytoplankton. And so I'm not sure what this tells me other than perhaps at that chlorophyll range, much of that chlorophyll is coming from pyridinium cells. And then as chlorophyll increases, you don't have an increase in pyridinium cells, but perhaps it's other stuff like pseudonychia and all the other various things that are floating around in the water column. So as a management tool, I mean, I think this is really informative. I'm just not sure how you can develop a target based on this relationship, because basically what you're showing, I think, is essentially autocorrelation, where you have, you know, how do I, if pyridinium is responsible for at least a chunk of the chlorophyll production, how do I manage chlorophyll? Like, how do I reduce chlorophyll without reducing pyridinium? So I think it's almost like the same thing that we're looking at here. And so along those lines, I think, I mean, just my opinion is I think maybe we shouldn't focus so much on chlorophyll. I think that's not really an endpoint. It's an indicator. That's a really important indicator, but as a target, I don't know if it's all that effective. I really loved the graph or the figure that you had on slide 15, which was really something I was hoping that you guys would show us. And that's this sort of divergence between this normalized load versus the actual load. I think that top right figure paints a completely different picture. And I suspected this was the case, but now you've shown that. And I think it's really important because it paints a very different picture of what's going on in Old Tampa Bay. And so I think maybe the first step, instead of struggling on, well, what chlorophyll target should we pick? Maybe the second bullet on that last slide is more relevant, which is, you know, how do we target nitrogen loads, assuming that is the only issue? And, you know, maybe it's the only one that we can control. So we know that this is something that we can deal with. Maybe that's the one that we really focus on up front. And then maybe look at another potential target, or maybe chlorophyll A as not a target so much as an indicator. I don't know if that makes any sense from the regulatory perspective, but you know, the problem also I have with chlorophyll A is just the idea that, you know, we were talking about alternative pathways for nitrogen assimilation. And so we talked a lot about today chlorophyll, pyridinium, seagrass, but then we've also got caulerpa to deal with, and then we've got drift algae to deal with. And I think there may be an avenue there to look at some alternative nitrogen pathways as well, as we start to kind of build a case to revise or maybe look at better targets. Anyway, those are my thoughts.
Yes, I think some of what you're expressing is just discomfort with the language that's sort of been ensconced in the paradigm itself. And so I would say that I do think that the direction basically that things are pointing right now is that we have a data deficiency to explore an alternative pathway into macroalgae like you're talking about, but we agree that that's something that's important.
Yeah.
But for the purpose of advancing this over the course of the next year, probably the biggest decision that needs to be made is whether or not the total nitrogen load to Old Tampa Bay stays the same and we
Right.
stop hydrologically normalizing it, which is going to be a big deal for folks in the nitrogen management consortium and folks in the stormwater side of things.
Yeah, definitely. And I do think that you make a really strong case with that figure, the chlorophyll pyridinium figure that, I mean, at the very least, it does appear that the chlorophyll target may be too high. And that I think this would argue that regardless of what that really means, a lower chlorophyll value is desirable in Old Tampa Bay. And I think you've shown that here.
Yeah, and I think then the next thing is we're showing already that we're not meeting the chlorophyll threshold in Old Tampa Bay regularly. So the question is, can we manage towards a particular chlorophyll endpoint that would produce related ecological benefits? And so that target that we're talking about, we're taking it sort of out of the realm of the nitrogen management consortium and into the realm of the TAC and saying, okay, so if we manage for a different algal community in Old Tampa Bay, then what are the management interventions that we do to get to that outcome? And so then we have these suite of modeling tools to look at, okay, if part of it is if we're no longer hydrologically normalizing the nitrogen load, that's, basically going to take everything down a notch to begin with. But then if there are further ways that we can target those nitrogen load reductions to both space and time that might amplify the benefits in terms of an algae response, those are the things we want to help you all prioritize as a management community. And then if that's not enough, do we need to then look at some of these other alternatives like hydrologic modifications or things like that? And then we need to sort of move on to the finding out phase of whether or not that does actually produce a positive seagrass response. And if it doesn't, then we need to get really serious about some of these things like the alternative pathways, macroalgae, and or whatever. Then we need to go back to the term that's in my mind, but y'all can figure it out.
Yeah. And just since I'll stop talking here in a second, but since I've got the mic, I'll just one last point to answer kind of the question that we were talking about on the last slide, that second sub bullet in the first bullet, which is do we want to look at this as an Old Tampa Bay segment wide issue, or do we want to make it more refined? I definitely would say steer away from WIBIDs, because sometimes unless we're comfortable with how those boundaries were drawn, I don't generally focus on the WIBID, but I do like that finer spatial scale where we have the northwest, northeast, the central, the south. Maybe not to that level, but I do think that there's clearly a difference between that northern section of Old Tampa Bay above the causeway, where you have a lot of those legacy sediments. It's much more organic, and it does appear, at least from some of the work that I've seen, that pyridinium, at least in its resting phase, tends to favor those organic sediments. So it does seem like that's the source. And I think Miles, I don't remember if you said it or not today, but somebody said that seems to be kind of where most of this is being driven. Yeah, I think Miles, you said that just recently, that that some of those, that figure with the different low, medium, and high cell counts, a lot of that was being driven by that northern area, and that makes sense, because that seems to be the more problematic area. So I would support breaking it down into a more finer spatial scale than just looking at Old Tampa Bay as a whole. All right, that's it. Thank you.
Real quick, Chris, we didn't put them up there because, like Miles said, the R-squared values weren't that great. They were less than 0.5, so it kind of puts it in the Is it just because there's enough samples? Is it because they were sampled by different entities that may not be on the same date?
Yeah.
But it was less than half of the correlate, there was a correlation where less than half of that chlorophyll A concentration was being explained by the pyridinium. So we looked into that, but it wasn't very earth shattering, I guess it goes, but
Yeah.
that's why that other graphic that showed the curve over time and kind of that drop off was more of the important takeaway on that one. And the other thing, just real quick, I don't even, one of the things that Miles had done in his in the background, which it wasn't ready yet, and I don't know how it would display, but he took all the data collected by FWC and basically did a animation of showing where those the different low, medium, and high, and having different circles showing like when they popped up, where the hits happened on these various locations. And did they pulse? Did they show up? Did they extend for a longer period of time? And probably the one interesting takeaway that we had is, I know you all started sampling in 2022, 23, kind of in the Feather Sound area, and that's, at least on that data, they seem to light up first and light up longer as far as those blooms on the animation over time. The other area was coming from Lake Tarpon. So it just kind of solidifies with some of this other data that we presented to you all, that the Northwest and the Central West seem to be the areas of most concern as far as pyridinium bloom initiation and duration.
The sediments off Feather Sound are pretty different than the sediments, up by Lake Tarpon.
Yeah.
But it's interesting that that's where they kind of lit up.
Yeah, well,
It's not really Lake Tarpon.
Yes.
The 54 square mile Brooker Creek watershed. Lake Tarpon is,
Yeah.
it just comes in and out. So it's really
It comes from Lake Tarpon and that whole other
the outfall,
eastern, yeah.
the Lake Tarpon outfall canal. The lake proper.
Sorry for the generalization, but that region, and yes, the watershed coming in on the east and the northeast is a lot larger than spatially than Tarpon. That northern input, I guess we should say, clarify is where that is.
I just had a comment about the chlorophyll and pyridinium graph. Just in the interpretation, I see the value in comparing the max to the means in terms of like evaluating chlorophyll targets, if that's on mean value, but just, I would interpret it a little differently as, because we do see when we're looking at point data that as pyridinium blooms get more intense or higher concentrations, it's really most of the it's not other, that's dominating the chlorophyll signal. And so just when you see that kind of leveling off at those higher levels of pyridinium or higher levels of chlorophyll, I guess, I wonder if that's because of the mismatch between comparing the maximum and the mean, because we know when we look at the data directly that we are getting that, there's extreme chlorophyll highs are related to extreme pyridinium highs. Just a comment about that. And then at the lower level, chlorophyll exceedances, that's often like a mixed bloom, not just pyridinium, or it could even be, diatoms that are contributing to the target, but not like those super high.
And this was the breakdown of all the different sub segments, what they look like. You can see that this is the Northwest, and that's where that signal comes up. And Miles did look at it both ways. He looked at them, and then I know that also does some more flattening, but the mean pyridinium abundances compared to the mean chlorophyll. And this, you saw the same picture, but it was muted. So
yeah, I think it's just, when you're comparing the means, because it becomes so sort of patchy, when you're looking, I'm just trying to make the point that, that those high chlorophyll events are pyridinium most, rather than a mixed community. That's just what I'm trying to think. So
Yeah, if I could address that, I think that's a good point, and it's what Chris said as well. So I'm not showing it here, but if you could imagine an additional curve, where because I was curious about this as well, as like dominance. Is there a chlorophyll level associated with like a switch in dominance or something? And we're not showing it here, but I did make a sort of alternative to this plot where there's an additional curve where it represents the pyro plus diatoms, which the Basilaria group, which is sort of typically the other sort of dominant component. Off the top of my head, I don't remember exactly what the story is there, but I think there were a few cases in some subsegments where you do see some sort of shift at a particular chlorophyll level, and that's super relevant and interesting. So that'll make it into the report, but that's a more recent thing that we added that I can't tell the full story today off the top of my head. But yeah, definitely something worth thinking about. And I do want to reiterate that on these plots, so like for any given value on the horizontal chlorophyll axis, the the curve and the band correspond to pyridinium at or below that chlorophyll level. Um, so for example, 9.3 is marked. So it's any months where chlorophyll on average was at or below 9.3. That's the corresponding distribution for pyro maxima. I also did it in terms of the median pyro value, and it doesn't really change the story much. The results are pretty congruent whether you use the median or the maximum for pyro. Um, just obviously max pyro is sort of the the more relevant in terms of outcomes and regulatory and management kind of considerations. But yeah, we're looking at the sort of both the maximum and the median. Um, and yeah, I'd like to kind of talk more with the group about this kind of thing, as we develop this. Um, Especially you, Chris. I've been wanting to have a chance to talk to you about this, so maybe we can get together sometime soon. Um,
Absolutely.
but again, it's just a sort of first pass. Just using history as a guide. And then to get to the point about like chlorophyll targets versus nitrogen and so on that Chris was talking about before. Yeah, this is sort of, it's like steps toward, ultimately like what are the levers we can pull, right? So like setting a different chlorophyll target isn't a direct intervention on its own. It's how do we get there? And since it's an existing, the chlorophyll target or threshold is sort of an existing um thing to manage for, then the question is, how do we adjust nitrogen loading? Is it enough to just consider actual instead of normalized loads? Another type of intervention we've talked about is like modifying the discharge regime from Lake Tarpon, especially considering that the northwest segment is particularly problematic with pyro. So it's sort of a stepping stone approach, and what this can do, this particular analysis maybe can feed into the logistic regression where we can estimate the probability of attaining a certain target or threshold for chlorophyll, and then under different conditions of temperature, nitrogen loading, and so on. But the question is, what threshold do we want to adopt on that sort of vertical axis of the logistic regression? So yeah, this is just kind of one step in that process, and there's other stuff as well. It's just ongoing. Um, there was something else I wanted to say, but it slipped my mind, so I'll leave it there. Thanks.
Um, so what if really it isn't one thing? It isn't, it seems like it's like, what if it is a combination, which is more than likely is we have legacy sediments that are a problem. It's not rocket scientist to anybody who's done benthic monitoring up there. We have in the two worst segments, we have 2 surface water discharges of reuse water. What is the timing of that? Obviously, we've been incredibly dry, so those discharges are probably non-existent at this point in time because they've been using every drop they have. But when we get into the wet season and it's not being used, it's, you've got a combination of stormwater discharges and surface water discharges of reuse. Part of that, then you've got the hydrologic issues as well. And so what if you just can't, managing to one of these targets really isn't going to get us. And it's more, it's bigger than that. It's a, you know, there's all these facets to it and we have to manage to something, to all the spokes in the wheel.
I mean, we were never on, at least as far as we evaluated the analysis, that it wasn't gonna be like one thing fix all. It's a combination. But then it's using the data that we have historically, potentially adding on some additional modeling to see what seems to be the best path forward. And like you said, is it a combination of, you know, non-hydrologically normalized loads? And then in conjunction with, does it need to look at seasonality? And how does that relate to chlorophyll? Because they are all interconnected. So that's, we're not under the impression that, you know, you can just tweak one lever. It's how do you manage each one the best way possible to give you the best outcome?
Are you gonna be able to kind of look at all those facets together? Or, I mean, I seem to be, I'm not a modeler, so it seems incredibly complicated to me, but it, you know, it, like you said, if you've got all these different levers and we've gotta figure out how to minimize impacts from each, you know, how are we gonna demonstrate? Because, you know, that's been part of the conversation with, there's been a lot of discussion and, you know, with trying to get support for modification of the causeways to support, you know, better tidal flushing, but the pushback was, well, there's no data to really show that that's a problem. So if we can't definitively show that these are the actions that we need to take in order to drive positive change, I don't wanna be caught in the same.
That's mostly the third task, and that's sort of Miles's specialty, and that's why it's beneficial to have, you know, him on the team. But I will say, from my read, if I was gonna boil everything down of what we've learned so far, is that the paradigm is still more or less true. We might need to think about it slightly differently and just target pyridinium. And we might be able to influence pyridinium to a point where we're getting back into compliance if we get real about further nitrogen load reductions, because that bay segment hasn't seen the same nitrogen load reductions as other bay segments. And so it's not anything earth shattering at this point. It's just the pill. But that third task that they're going to be working on is basically asking and answering that question of, okay, so if we ratchet the actual nitrogen loads down, do we think this will produce outcomes that are beneficial, not just to seagrasses as an endpoint, but to pyridinium and chlorophyll as an endpoint? If the answer is no, then that is useful data that says, okay, then we need the yes. I think we need a yes and strategy anyways, but that's like a really strong case for the yes and strategy and advancing some of these, you know, complementary management actions that we've been talking about. But right now we haven't been able to sort of rule out that there's not more to be done on the nitrogen load side of things.
Any other questions at this point? Yes.
This kind of follows up a little bit with Ellie's comment. So I was really struck by Miles' plot of the regularity, the annual cycle of these blooms. And bring it up. That occurs, right? So that is occurring in the context of, you know, strong variability year to year of rainfall and commensurate stormwater and non-point runoff and all these hydrologic sources of nutrients. But yet we're getting this pretty regular cycle. So does that, I'm just throwing this out there, right? Does that mean that those are not particularly good levers to use if we want to affect these blooms? And we need to find something else, whether it's doing things that people don't want to do with the sediments or something else, maybe.
I'm not dredging old Tampa Bay until we get our nitrogen loads under control.
Well, I mean, if you're not that, I mean, this says it doesn't matter. This tells me it doesn't matter what those loads are.
If you're,
Whether it's
if you're gonna
dry or
continue to put that same input, I'm not committing this region to dredging old Tampa Bay at regular intervals, so long as we continue to contribute those you know, sources of mucky sediments and those nutrients. Like if we don't make the upstream changes, we're not gonna go down a path of that kind of large scale management intervention. That doesn't make sense to me.
And you're seeing those patterns because things are the way they are. I mean, you have the loading and, you know, it's like the normalized load, it looks great, but then you have the non-hydrologically normalized load and I don't think miles has mirrored it on top of each other, but I would suspect you might see a similar situation of those peaks if there's not some sort of delay.
Steve's point's a good one. I mean, it struck me. I gotta say, like when it's not showing on this plot exactly, but these data in the top panel here, I put them through signal processing, and like 90% of the variability is explained by what's essentially a seasonal cycle and a couple other components. That's really striking because when I've done this kind of work before, I mean, it's generally like as you go up levels of organization from, you know, physics to chemistry to biology, you get weaker signals. So 90% for something biological like this is really striking. And underneath that, like Steve pointed out, there's a lot more variability with discharge and hydrologic loading, nitrogen loading and stuff. However, I don't think that necessarily means that all that variability underneath it, you know, doesn't explain it. And I mean, that's a question. I think that's something that we want to investigate. That's something I hope to have done before our draft report in July. You know, that's a more complicated question. And, you know, that's a hypothesis, Steve, and it might turn out to be, you know, right. But I think that's something we've got to investigate because um I think, you know, there's going to be some causal relationship there that we can uncover empirically. Um, but then, you know, we want to compare and see, like Maya is saying, like, you know, it to what extent is that a lever? And uh, yeah. So anyway, that's something that we want to approach because it is an important question. Um, and internal loading is something we've talked about in the past. It's kind of hard to get at because the data on that, you know, obviously um, aren't really as good as things like external loading. But we are thinking about that, especially in terms of modeling.
I hate to end this discussion. I think it's really good, but Justin drove all the way from Pensacola, so I want to make sure he gets his time. But yeah, this is the last presentation for today. He's got some really cool data to show us.
And I know that we're well over time, so I will try and go quickly, and this is being recorded, right? So if you gotta bounce or drop off, I totally understand. Little bit different than the other talks we've heard this morning. Nate is an economist up at our Narragansett, Rhode Island lab, and I'm a social scientist up at our Gulf Breeze, Pensacola, Florida lab. So all of our work kind of sits at the intersection of people and the environment, and this is no exception. We're generally talking about trying to get a count of number of recreators for a given space. So why does the EPA or why does anybody really care about this? Is this just an excuse for you guys to go sit at the beach all day? Well, recreation is one of the ways that we are able to connect all of the things that we've been talking about today with water quality to people and how people are interacting with it. So if you can show that a given improvement in water quality means that more people go there to use that water body, spend more money on their vacation or just enjoy their trip to the beach more, you can take that, relate it to the water quality improvement or difference in water quality and start to be able to assign different values to that improvement. So non-market values. So these are things that, you know, when you're going to the beach, you're not paying $10 today because water quality is better. There's less nitrogen in the water, let's say. But your behavior might demonstrate how you value that. So maybe The beach that's just down the road from you has a lot of closures, and you have a perception that it's dirty, so you drive a little farther, a few miles farther to one that's down the way, that you have a perception of higher water quality there. And we can take that difference in driving distance and driving time, that it was worth it for you to expend to get that increased water quality, and again, assign that value over to it. And we use this in policy decisions, because any new rulemaking, any new regulation has to go through rigorous cost benefit analysis. We have to show that, say, a TMDL is cost effective. And this is one of the ways that we are able to put values in that benefit column. So just to give a couple of examples of ways we've done this in the past with kind of existing information. The first is a paper that Serena wrote that was looking at the costs of closures up in the New England area. And basically using benefit transfer, being able to say, okay, a day of being closed, how much is that costing us? Right? What's the lost value with people not being able to go there to recreate in the same way? And then the second paper here is by Julia, and was identifying disparities in equity. So places where the demographics or the income was lower, we were able to show a correlation with lower water quality. That was focused in Rhode Island. So to do this kind of work, to kind of move it in the direction of getting it better, more accurate, here are some of our data needs. So we really need more information on people's use of these natural areas. That includes visitation. So how often are they going? How long are they staying there? Where they're coming from. Remember, I gave that example of you go a couple miles further down the street. Well, it makes a difference how far you went to get there, because that's kind of what you had to put into the transaction to get the benefit. And then, of course, also any information on demographics that we can gather, particularly around EJ issues with access to these natural areas. Now, there's an existing body of literature on this. This has been done for a long time, and we have kind of traditional methods. We can go out and do intercept surveys where we stop people and we ask them where they drove there from. We ask them about expenditures, all of those types of things. We can also go and sit and count people. But there's limitations of these. There's a lot of logistics, there's a lot of expense to that. And it's very hard to scale across either space or time. It's also hard to use those, leverage those as we get better, higher resolution information on kind of environmental quality, right? So we're getting better water quality information that we can really differentiate between that site down the street versus a few miles down the road. We really want to be able to differentiate the visitation to those as well. So a little bit more on data sources and kind of focusing in on what I'm gonna be talking about. I could probably give a talk entirely on any of these data points. But today we're gonna focus on the human behavior side of the equation, and less on the traditional surveys and on-site observations, and more on this newer human mobility data. And that's really a combination of traffic sensors, social media, as well as cell device locational information, which is what I'm gonna focus on. So cell phone data, mobile device, you've probably seen examples of this in the literature, how it's being used for a variety of different things. No surprise that we're trying to use it here as well. And this top box represents what the vendor is doing. So we're not, you know, if you, for example, my wife really likes ID shows, right? The crime forensics shows. And in a lot of those, you'll hear where, oh, they got the cell data and they figured out where the towers were and did a trajectory to figure out where they were during the time of the murder, right? That's not what we're doing here. That's not even what these vendors are doing here. A lot of times, especially now, when you sign up for a given app, maybe it's a weather app or something like that, when you're scrolling through to agree to the license agreement, a lot of times part of that is that they'll be able to take any of your locational information that's collected while you're using the app and can sell it for marketing or a variety of different things. So what's going on here is they're selling that information to the vendor. The vendor is buying it from a bunch of different sources. It's all pre-aggregated, so it's not identifiable to you. And basically what the vendor is providing us is we say, hey, we're interested in this geography for this location. They query what they have in their database. They say, okay, here's the number of devices for that time period. Here's the duration that we estimate they were there for. And then here's the number of people we feel that represents. So they have a kind of black box model that, you know, they have built based on knowing where their data is coming from and how representative it is of the population. So they gave us those multiple pieces of information, but we're not interacting with that data itself. And even the vendor that we're buying it from, it's already pre-aggregated and anonymized. So then what are we doing on our side? Well, we're defining the query. So basically the area for that beach and when we want the sample. We get those results, and then what we do is we try and calibrate that to improve it for these types of blue water access sites. So, you know, the vendor has done a lot of calibration on their estimate already. A lot of times it's where you have a lot of people like a mall or large venues where you already have a known count that they can relate it to and calibrate to. But we know that these water access sites are going to be a little bit different. We might have some mismatches. So from that, once we've kind of recalibrated it, we are creating more sophisticated models to focus on kind of our outputs of interest, how these counts, how these visits, number of visits vary over time and vary related to other factors, particularly over space and particularly as it relates to water quality and closures. So let's walk through a quick example. You can take a beach. This is a beach up on Cape Cod. I believe it's Marconi Beach. We can focus in on a given geometry. This is what we'd send up for the query. We'd say we want results from this date to that date. Back hourly counts, device counts. So that's where you have a device total over to the right, as well as an estimate of the number of visits that number of devices represents. Again, these are broken out by hourly, so that's what the HH001 and so on represent. The date, you know, when we're sending multiple points of interest, the POI for that so that we can identify it. Also, they have a sense for each of these devices of the home block group. So again, it's anonymized, but they do know based on the amount of time spent during certain hours. And I believe it's looking at the past month where they're kind of coming from. So generalized to the census block group, which for us is great because we can get census ACS data at the block group level, which gives us a sense at least of demographics. So that data is great, but again, it's an estimate. So we need some on the ground, ground truthing. So two things here. One is paper, writing up basically us going out and doing those counts at a bunch of points of interest in Cape Cod. On the right, on the right here is an EPA report on the methods used for these different counts. And really, why I bring it up is how we're using it is to calibrate, okay, how good are these cell-based records? The black line, if you can see it, would be if there was perfect alignment. And you can see some of the smaller sites maybe do a little bit better, but most of these are overestimates based on just the cell phone data alone. Now, we can use that with a pretty simple regression-based model to calibrate the cell phone data better to these hourly visits that were observed. Now, there can be a bunch of reasons why it's so far off. So we are doing some work to improve these. But if you take nothing else away from this, it's okay, maybe don't take these estimates at face value. A little bit more ground truthing needs to be done. Now, once we have that calibration model, we can take it and apply it to other sites where we haven't done these counts necessarily. So here we have them applied for the entire summer of 2017, all of the sites that we have on Cape Cod. And you can see where green ones have a little less visitation, whereas the red have more, and it's not spatially weighted. So some of those red sites are just larger sites. We can also take it and kind of look back in the data for dates when we know there were closures. So when we did that on Cape Cod, there was a bit of a correlation. So we saw a decrease in visits by 16 to 25% on days when those beaches were closed to swimming. Now, it's possible that a lot of people on those days are still going out and are disregarding the advisory and are still going in the water. It's also possible that remaining 75% of the people that normally visit weren't going in the water anyway. We did find, however, that closures outside of the Cape, so when we expanded this to more of Massachusetts and Rhode Island, there was less of a signal. So the other part of the data that I mentioned was the home origins. So these are all of the block groups for a given site in Narragansett, Rhode Island, which I believe is Narragansett Town Beach. And you can see of the devices that they detected, these are all of the different places that people came from. Now, using this, we can estimate a travel time and distance. And we can take that and, excuse me, look at the income for those, average income for those block groups, apply a millage for the mileage, and come up with, okay, a dollar value for each of those trips to estimate a non-market value of something like $37 per visit. Now, obviously, for most of these visits that were really close, basically anywhere in Rhode Island is close to anywhere in Rhode Island, the value for those would have been lower. This is just when it's aggregated, what you have, that's an average. And when you aggregate it, it's 2.2 million for the month of July. So you can see where this really can heavily influence that kind of benefit side of a benefit cost analysis. So that's what's been done in the past. What we're trying to do is take these efforts and scale them. So we've expanded out from just Cape Cod and New England, and really have looked at, I think, over 800 sites now in the New England area. Again, going through all of this information, also bringing in water quality to the equation. Another effort was looking at using this data in national parks and other public lands. And there's a paper there. Basically, as these locations, national parks, a lot of times have standard visitor counts if there's like an entry fee, right? Or if there is some kind of gatekeeping mechanism. Now, in addition to expanding the scale, the spatial scale in New England, we're also trying to take this national. So in the Gulf of Mexico, what that means is we've been looking at EPA's Beach Advisory Closing Online Notification System, Beacon, which is housed in Office of Water alongside their beaches program. So they fund a lot of beach monitoring by the states. And basically what they've done is taken those water monitoring sites, sampling sites that they have, associated them with hydrologic lines, reach access data set lines for basically just over 600 sites in where they have monitoring data. Now, for their purposes, they tier those sites. Some of them are what they define as jurisdictional, so of particular interest to EPA based on where they are and the current monitoring program. Here is just an example of the sites that we are looking at here in the Tampa Bay area. So of those 612 beacon sites, we defined areas for all of them, not only based on the reach access data set, but also looking at NOAA's continuously updated shoreline product cost. And with these, we said, well, we can't afford to query all 600 at this point, but let's take a random sample of 91. And then let's kind of pick and choose a few extra areas to add that we know we might be more interested in the future. So this table is just information from the Beacon site to give you an idea of some of the information that Office of Water already has collected from the jurisdictions on all of these sites, and also to give you an idea of maybe what it's called locally. So for example, Treasure Island Beach is over in Treasure Island. One of them that I will talk a little bit more about is this one here in St. Pete, which is North Shore. And in there, it's as North Beach. So these were the three that we added. Gandy Boulevard, based on where it is in Old Bay. Part of what we're interested in with closures is, of course, contact uses, people actually going in the water, versus some sites that maybe have armored shoreline, and maybe people go there fishing and to kind of sit and enjoy the views, but aren't really having contact uses. So that was part of why we added Gandy Boulevard, as well as Bahia Beach and Island. And the second two are tier ones as far as Office of Water is concerned. All three in the Bay, because we are more interested in these interior sites that are probably used more by locals versus people that are coming from way out of town. Because we are more interested in day trips versus a lot of the bigger valuation studies that are done for tourism and marketing are looking at, what did you pay for your lodging during your time, those types of things. We're less interested in that. So here's that North Shore Park that I had mentioned earlier. This is the geometry that we purchased the data for. We are working on ways to improve that geometry to make sure that we're not getting visits that aren't really to the site that we're interested in. So you'll notice some weird cutouts here. Those are from roads and building footprints a lot of times. Not necessarily here, but for example, I had shown Bahia Beach. You have a little restaurant basically in this site on the northern end. So something like that, well, now you're still picking up those cell phones, so that might be a source of some of the overestimation that we're seeing. So as much as we can, we're trying to edit down these geometries so that we're just getting the results for the area that we're actually interested in. And this is an iterative process. You see some of these polygons off to the west here that in the newest version of the data set have been removed. But really why I wanted to pull this up is that this green line is how it's defined in Beacon. And had we had some on the ground knowledge when we picked it out, we might have, and had we picked it out, because remember this was in our random sample, we probably would have picked this green line just to the south. Because what goes on is, I believe you have some mangroves here and some armoring right here that you can kind of see in this image. And basically, this bit of water here is what you see in this imagery right at the southern extent of the site. So, you know, ideal world, we would have picked the one to the south, just to give you an idea of some of the things that we're working through, where this kind of southern beach area, and then down the way, there's some beach volleyball courts set up. That's, you know, what we're more focused on. So to give you an idea of what we saw in that data, so we queried it from 2022 to 1 month into 2023, because at the time of the purchase, that was the latest and greatest data available from the vendor. And if we looked at those number of devices versus the average estimate, we were saying about each one device accounting for almost 40 people. So this was definitely on the lower end of the accuracy. Usually it was 20 to, I think the highest was in the 40s. So definitely not one of the ones where the vendor had the best data. And to give you an idea of what we're doing now, when we go and we try and ground truth these, we're getting hourly counts of total number of visitors. We're also looking at what they're doing so that we can get a distribution, an idea of how many people are passing through the site quickly. Maybe they're just biking, walking along, or are they maybe sitting there waiting for a while, going in and out of the water, those types of things. So getting an idea of how the sites are being used, both so that we can use it on the valuation side when we're looking at contact uses, how are people using this water, and does that expose them to certain human health concerns, as well as can we use these differences in activities to further characterize and calibrate, better subset our sites, basically. So can we say that we should be calibrating boat launch in one way, whereas we should be calibrating coastal beach in another way? Seems pretty obvious that we should, right? So that's what I have for now. One thing I will say, if you have any of this type of information, any types of counts, whether it be lifeguard counts, parking data, or you know somebody that has this type of information, we don't want to be going out and doing the counts ourselves. If there's anything else out there that we can leverage, That's definitely what we've done up in the New England area and in the other bay systems within the Gulf of Mexico that we're working in. So if you have any of that, my email is up there. I usually joke if you can figure out how to spell my last name, you can find me. Because it's fairly unique. Just last name, first name at epa.gov. And I've also included our website for this research. If you want to go there and find any of the papers that were listed. I have time for questions, but I know we're.
Any questions for Justin?
I would be really interested to know, you know, because you're able to extrapolate this to cost as well. You know, what was the cost of the Piney Point outflow a couple of summers ago? And we know that the ecosystem suffered a lot. I think the economy of particularly St. Petersburg area did too. I would just really be interested to know what that was.
Yeah, I know NOAA has done a lot of work in that area and has some better monitoring for it. I'm not aware of any cost estimates that I've seen come through, but no, they've set up some baseline data for that, right? You know, that's the other place where this is often useful, particularly for NOAA's natural resource NRDA damage assessments, because if it's something like an oil spill or other kind of finable impairments, so to speak, you have to use that benefit that's lost to inform what the fine is. But that's an area that NOAA is definitely very interested in seeing this work progress for, because you don't necessarily know where there's going to be an incident like that ahead of time to get your counts to then say, well, we see because of it, there's half as many people. With our closures, we see because of the closures, it seems like there's a decrease in 16 to 25% of visitors. If you don't know what it was before, you can't do that. And it's really hard to go out and figure out, okay, how many people were using this site after the fact. Sorry, I know I didn't really answer your question directly, but I agree 100% where like that's an area where something like this is really useful.
So you may not be able to answer this, but how much does it cost to get the vendor information? How realistic is it to, you know, kind of do this on a periodic basis, or is this something that you can only afford to do once in a while?
So my favorite answer, it depends. We've seen the cost go down. We've been doing this for not quite a decade, but 7 or 8 years now, and we've seen the cost go way down, where initially we were pressed to get a handful of sites. We upped that to the hundreds, and now we're doing it at the thousands. It's hard to say how representative the price that we're paying for it is, because we're doing it in bulk and because we're the government, and a lot of times there's a little bit of a price increase with having to deal with the contracted limits, right? To really generalize, definitely under $1,000 per site for a year of daily visits. I think for around 1,000, yeah, we were right around that 10,000 mark. We're currently negotiating new contracts with this. We're hoping that that number is gonna go down and that because of an increased quantity with scaling it to national scales, we'll be able to kind of do it as an indefinite quantity. So it's as fast as we can send the queries and work through the data. That's the plan in terms of like what we're doing, right? Because with that, we'd then be able to make it more sustainable year to year. Because the idea is to build this into tools that can be used.
Justin, we're 5 minutes over, even though we started 30 minutes late. I won't hold it against you.
Everybody agreed.
Just two quick announcements before we wrap up. So just real quickly, we do have our Bay Mini Grants RFP out on the streets right now. It's our mini grant program that's funded by our specialty licensing tag, Urban Tag. So if you know anybody that's interested in that, please consider submitting, contact Jessica at our office. I believe that closes September 18th. So you got some time, but that's now on the streets. Second thing I wanted to mention, and I brought this up last time, about this time every year, we make a call for you all to provide potential special study sites for our benthic monitoring. So EPC does this every year. They've been doing it for decades now. But we get 20 sites each year where we can choose where we want to do some special studies. So this includes biological monitoring as well as contaminant monitoring. And last TAC meeting, I threw a call out there to say if anybody had any ideas. And I get a lot of great ideas from you all, but we ended up choosing some sites in McKay Bay and Palm River. And that's primarily because of a need to understand the potential effects of sediment contaminants on fish in those bay and people that are catching those fish and eating those fish, trying to see if there's a linkage there. So we're going to go with that. Probably going to be 12 sites, McKay Bay, 8 Palm River. Now's your chance to argue against that, but if we don't hear anything from y'all, that's what we're gonna do. So, is there anything else on that, Chris, you wanted to mention, or?
No, yeah, you covered it. We got an inquiry about the sediment contaminations in the fish tissues and stuff like that. So that's what was.
Hey, Chris, are y'all thinking about kind of doing a follow-up to the, I don't know, maybe it was like 2008-ish when Gerald and Ed did the fish tissue study that way? Like, are you gonna look at the actual bioaccumulation of the food?
Well, we would have to partner with something with FWC or something like that to do, because we don't do fish tissue, we don't do tissues, so.
That's like a kind of an important way of answering that
Yeah.
question.
So, right.
Let's maybe Carrie talk.
One thing we are doing right now is we started adding metals to our water column, our monthly water column samples that we're taking. So we can compare that at least to the sediments, even though they're two different matrices, but so yeah, the tissues would be another step.
Okay.
Any other announcements? I have one, our sister program down in Sarasota Bay, their senior scientist, Jay Leveroni, is going to be retiring later this year. And so keep your eye out for that senior scientist position getting advertised soon. And if you know anybody who might be interested and be a good fit, encourage them to apply.
There's the Clyde Butcher Living Waters photography exhibit at the Clearwater Historical Society ongoing now. And this is to share information about aquatic preserves around.
Okay. Thank you all for coming. Apologies for the difficulties with getting it. It won't happen again. I'll take full responsibility for that. You made it happen. Appreciate y'all.
That was a kismet of things happening.